Hello everyone, welcome to SS Classroom. So now today we will discuss the transportation of molecules across the cell membrane. In the previous lecture we discussed the meiosis and today discuss the transportation of molecules across the cell membrane. Means how the extracellular fluid substances transfer inside the cell and how the cell material or cell molecules transfer from cells to the outside or extracellular fluids. So we will discuss in this lecture. So what are the key points we will cover? Passive transport and active transport. So there are two transport occurs across the cell membrane. First is the passive transport and second is the active transport. So what are the difference between passive transport and active transport? Passive transport, transfer of molecules across the cell membrane without the use of any type of energy. But active transport, transportation of the molecules across the cell membrane with the help of the energy in the form of the ATP. So you can see passive transport, there is no need of the energy when transfer of the molecules across the cell membrane. What are the types of the transport? First is the diffusion, next is the osmosis, and next is the facilitated diffusion. Now, what are the active transport which require the energy? Exocytosis and endocytosis. And again endocytosis divided into two stages, phagocytosis and pinocytosis. So we will discuss in a detail. So first of all discuss the passive transport. What will happen in the passive transport? In the passive transport we will discuss the diffusion. So already we discussed the meaning of passive transport. Passive transport means movement of molecules across the cell membrane without the use of any type of energy. You can see. So first is the diffusion. So diffusion means transfer of molecules from the higher concentration to a lower concentration and cross the cell membrane without use of any type of energy. You can see. This is the plasma membrane. Here is the low concentration inside the cell, and in the ECF or extracellular fluid there is high concentration of molecules. Then higher concentration transfer to the inside lower concentration. So movement of molecules, you can see movement of molecules from the higher concentration to the lower concentration. So this process is called a diffusion, passive transportation diffusion. Next is the passive transport diffusion, facilitated diffusion. Facilitated diffusion means a special type of facility provided in the plasma membrane in the form of the different types of proteins. For example, this is the channel protein. You can see this is the channel protein and this is the carrier protein. Channel protein and carrier protein. In the channel protein there is tunnel. You can see there is the tunnel or channel, and these are the molecule transfer from extracellular fluid to the inside in the cytoplasm without the use of any energy, but there is special types of facility provided in the form of the different types of the protein. Here is the channel protein. So this is called the facilitated diffusion. And the another protein is carrier protein. So you can see this is carrier protein and this is plasma membrane. This one extracellular fluid and this is intracellular fluid. When the special types of molecules attach with the protein, then protein change into the structures and transfer the molecules from the external environment to the internal environments. So these are the special type of protein present in the plasma membrane, and completion of the special type of diffusion, that is called facilitated diffusion. So in the facilitated diffusion, the movement of molecules across the cell membrane from the higher concentration to the lower concentration with the help of a specific transportation protein that is called the carrier protein and channel protein. So this one is the channel protein and this is the carrier protein. So this is called the facilitated diffusion. And what happened in the osmosis? Osmosis is the just opposite of the diffusion. In these conditions, the movement of the molecules from the lower concentration to the higher concentration. Or we can say there is osmosis, the net movement of solvent molecules. Means movement of the solvents, usually water, across a selectively permeable membrane from an area of high water potential to low solute concentration to an area of low water potential means high solute concentration. Means movement of molecules from the lower concentration to the higher concentration. But movement of the solvent is higher concentration and movement to the lower water concentration. So movement of the solvent from the higher concentration to the lower concentration. But movement of the molecules from the lower concentration to the higher concentration, this is called osmosis. So you can see this is the image and movement of the solvent inside the cell and enlargement or swelling of the cell. This is called osmosis. Next is the types of solutions. So there are different types of solution. First is the hypotonic, next is the isotonic, and third is the hypertonic. What are the difference between hypotonic, isotonic, and hypertonic? Hypotonic means lower solute concentration and higher water concentration. How the formation of the solution? First is the solute molecules, then solvents, and formation of solution. In the two things, first is the solid molecules, second is the solvent. Then what will happen in the hypotonic solution? Solute concentration is lower and water concentration is higher compared to the another solution which may be present toward the cell. For example of the solution that is called 0.45 normal saline. So normal saline generally present in the hospital and 0.45 normal saline that is called hypotonic. 0.9% normal saline also present in the hospital settings of clinical areas that is called the isotonic. But 0.45% normal saline, another name also called half normal saline. So half normal saline 500 ml NS comes under the hypotonic solution because of the lower solute concentration but higher the water concentration. Next solution is, you can see this is the hypotonic solution. So hypotonic solution means decrease the solute molecules and increases the solvent. Next is the isotonic solution. Isotonic means equal. Solute and solvent concentration are the equal. If the concentration is equal inside the cell and outside the cell, there is no differentiate in the concentration of the solute and solvent, then there is no movement. There is no movement of the molecules from one concentration to the another concentration. Why? Because there is no changes in the solvent and solute. There is no higher concentration of the solute and there is no higher concentration of the solvent. Both are the equal concentration. In this condition, there is no transfer of the molecules across the cell membrane, inside of the cell and outside the cell. There is no movement. That is called the isotonic solution. What are the example of the isotonic solution? 0.9% normal saline and ringal lactate 500 ml. You can see easily in the clinical setting. You can see this is the isotonic solution. So isotonic solution, water going inside and molecules traveling outside, but there is no changes in the cell structure. You can see in the first structure, solvent go into the cell, then enlargement of the cell. You can see enlargement of the cell when the solvent inside the cell. But you can see isotonic solution, movement of molecules, movement of solvent, but there is no changes in the cell structures because there is no movement of solute and solvent because of the equal concentration. Equal concentration present inside the cell and equal concentration present outside the cell. Outside the cell in the extracellular fluid and inside the cell in the cytoplasm. That is called the isotonic solution. Next is the hypertonic solution. Hypertonic solution means higher the concentration of solute molecules and lower the concentration of solvents. Solvent means water compared to the another solution. Because of the comparison need the two solution for the comparison. So one solution inside the cell that is called cytoplasm and another solution is extracellular fluid which is outside of the cell. So what is the example of hypertonic solution? 3% normal saline, 5% normal saline, and 10% dextrose. Another also, but these are the some example of the hypertonic solution. So what is the meaning of hypertonic solution? Higher the concentration of the solute compared than the solvent. Then what will happen? Burst or narrowing of the constriction of the cell because of the higher concentration of the solute. Then movement of the solvent outside the cell. You can see this is the cell and this is the extracellular fluid, and extracellular fluid has higher concentration. Higher concentration fluid is present is 3% NS, it may be 5% NS or dextrose solution. So this is the dextrose solution. Outside is higher concentration and cell doesn't have a higher concentration of the same type of solution. Then from the inside of the solvent is present, solvent means water, and water move to the higher concentration from the cell to the extracellular fluid. So this is called narrowing or constrictions of the cell. You can see constriction of the cells. So these are the three types of the solution. One is the hypotonic solution. Solvent moves inside the cell. This is the isotonic solution. There is no movement or there is maybe little bit movement may be present, but there is no changes in the cell structures. Why? Because of the equal concentration inside of the cell and outside of the cell. Inside is the cytoplasm and outside is the extracellular fluids. In the hypertonic solution, solution is hypertonic, but solvent inside the cell is lower concentration. Then solvent from the cell moving toward the hypertonic solution. Then constrictions of the cell occurs and swelling of the body is reduces. So these are the three types of solution. I hope you understand. Next we discuss the active transport. So active transport means the transport of substances or molecules across the cell membrane with the help of the energy in the form of the ATP. You can see active transport means transfer of molecules from the lower concentration to the higher concentration. But there is no possible of transfer of molecules from lower concentration to higher concentration without use of energy. That's why use of the energy in the form of the ATP to transfer of molecules from the lower concentration to the higher concentration. From the extracellular fluid to the inside of the fluid and inside of the fluid to the extracellular fluid. For example is sodium potassium pump. Sodium potassium pump means transfer of the molecule sodium and potassium inside and outside of the cell. So what is the meaning of active transport? It is the moving of molecules across the cell membrane from any area of lower concentration to an area of higher concentration, which is against the concentration gradient and requires cellular energy. Cellular energy means require the energy in the form of ATP. What are the example of active transport? Exocytosis and endocytosis. Exocytosis means movement of molecules from the cell toward the outside of the cell. And endocytosis means movement of molecules or substances from the outside of the environment, that is extracellular fluid, to the inside of the cell, that is cytoplasm. So first of all, we discuss the exocytosis. Exocytosis means toward outside of the cell, means movement of the molecules or substances from the cell to the outside of the cell. You can see exocytosis process. So this is the vesicles and transfer from inner environment to the external environment. You can see this is the attachment or fusion with the plasma membrane and release of the molecules from the inner environment to the external environments. So this process of the plasma membrane is called a exocytosis. So exocytosis release the substances from the inside of the cell to the external environment with the help of the energy. Next is the endocytosis. Endocytosis means toward the inside of the cell. Inside of the cell means the movement of molecules or substances from the outer environment to the inner environment of the cell. Endocytosis is the cellular process where the cell membrane engulfs and internalizes substances such as nutrients, fluid, or even microorganisms from its external environment by forming a membrane-bound vesicles. And there are two processes of the endocytosis. First is the phagocytosis. Phagocytosis means cell eating. Cell eating the different types of bacteria inside the cell, and there is a special type of enzyme, that is lysosome, that is hydrolytic enzyme, neutralize the bacteria and destroy the bacteria inside the cell. Next is the pinocytosis. Pinocytosis means cell drinking. When the cell is hungry, then taking the water molecules in the large amount inside the cell by the process of pinocytosis. So you can see phagocytosis for the large amount of the molecules or large size of the molecules, and pinocytosis process for the small size of the molecules. So you can see this is the phagocytosis process. This is a bacteria and cell change into the structure and taking the bacteria inside the cells and neutralize the bacteria by help of the lysosome enzyme. So this process done by a special cell that is called the phagocytes or WBCs. You can see pinocytosis. Pinocytosis means different types of water molecules comes and formation of the vesicles, and these vesicles go into the cell with the attachment of the plasma membrane. And formation of the vesicles, you can see, and movement of the vesicles inside the cell when cell is drinking. So these are the process of the active transport and passive transport of the molecules across the cell membrane. I hope you understand. So this is all about the transportation of molecules across the cell membrane. We discussed the active transport and passive transport. Passive transport means diffusion, osmosis, and facilitated diffusion done by the special types of protein, that is carrier protein and channel protein. And active transport done by the phagocytosis and endocytosis, means exocytosis and endocytosis. Endocytosis again divided into two, phagocytosis and pinocytosis. In next lecture, we will discuss the different types of tissue, that is epithelial tissue, connective tissue, muscle tissue, and nervous tissue. So we will discuss later. Till then, thank you so much and thank you for watching video. Thank you so much.